Today I'm here with Dr. Sabrina Arena at the Institute of Cancer Research and Treatment in Turin. Dr. Arena, you're here at the Congress talking about efficient tumor therapies based on the characteristics of tumor cells. Most people are familiar with bacterial cell resistance to antibiotics. However, what you're talking about is cancer cell resistance to targeted therapies. What all is known about this mechanism and how common is it?
Unfortunately, this is a very common mechanism. This can actually arise in primary resistance because many patients can harbor mutations that make them resistant to these therapies. Or actually, those patients that have initially responded to these target therapies can eventually develop secondary resistance due to the emergence of novel genetic mechanism of resistance.
What all do we know about the mechanism behind this resistance?
Well, about this has been done a lot of research trying to understand which are these mechanisms at the molecular level. In order to understand this mechanism, many preclinical models have been developed in order to test and design novel lines of therapies to treat these patients.
So what are those? Some of those potential solutions?
Well, the first solution would be to try to understand which genetic alteration, which mechanism of resistance the patient is developing during treatment with target therapies. And in order to do that, we have taken advantage of a novel technology that is named liquid biopsy. So basically, we can harvest blood from patients that are under treatment with targeted therapies, and then we can interrogate from this blood for the presence of any genetic alteration that have been emerging during the treatment with target therapies. So whenever we can monitor and understand which are the mechanism of resistance arising, we can understand which is the best therapy to use at that time in order to fight in that moment the disease.
So how do you see this approach helping to limit the emergence of resistance and lead to long-term response in patients? But also, what is the pharmacist's role in this therapy?
So catching the mechanism of resistance in advance could help us to really understand which will be the most effective therapies against those mechanisms of resistance that are arising. In this way, I see the pharmacists really like those people that can bridge the gap between the molecular biologist that has identified the molecular mechanism of resistance arising in patients and the clinician that will have to decide which is the best therapy to be used. So the pharmacist can try to understand which will be the best drug to be used for the right patients at the right time, the right moment, with the right formulation, in order to avoid also toxicities and to make this treatment as effective as possible.
Fantastic. Thank you so much. This is Whitley Yee at the FIP Congress of Pharmacy and Pharmaceutical Sciences in Glasgow.